Welcome. This presentation is going to look at some of the quantitative aspects of a couple of topics that we've been looking at as our foundation of knowledge kind of builds on topics related to this specific integrated STEM project that we're kind of building up towards. So this is going to look at something called FRET, or Forster resonance energy transfer, within the context of photosynthesis, and then also ways that we can measure photosynthesis. So we'll begin initially looking at FRET and what FRET is. So FRET deals with fluorescence molecules, and as you know, fluorescent molecules have to be excited by a photon of one wavelength, and then they emit a photon of a different wavelength. Right? And that's because there's some energy lost within this process that's lost as vibrational energy or heat. So the wavelengths are going to be longer after they've been absorbed and then emitted because there is that loss of energy. Now, if there's two fluorescence molecules and one excites at a specific wavelength and then emits at another wavelength, that can be coupled to the excitation of a second fluorophore or fluorescence molecule as long as its excitation is the same as the initial molecule's emission, right? So a visual of that might be helpful. So if we look at the first fluorophore here, right, so it's getting excited by blue, and then it emits a green light, so that's of a longer wavelength, less energy. There could potentially, depending on context, be another molecule that can get then excited by that energy, right? And that'll bounce its electron into a higher energy state, which will ultimately fall down to the ground state and in turn emit a photon of a wavelength of an even longer distance, right? Or an even lower energy. So in this way, with these two coupled fluorophores, we could theoretically excite with blue light and then observe or see red light. So there needs to be an appropriate overlap with fluorophore 1's emission and fluorophore 2's excitation. And they also need to be within a specific distance, right? So that's shown here as 1 to 10 nanometers. So they need to be within proximity to each other for this transfer to occur. So now to put this in the context of photosynthesis, as a reminder, right, photosynthetic organisms basically need, well, quite a lot, but some of the basics are molecules that can absorb a photon, and then some sort of a membrane to be able to disproportionately localize, in the context of photosynthetic organisms, hydrogen ions, which can then drive the synthesis of ATP. It's a little more complicated than that, but those are some of the fundamental kind of parts that you need for the machine to make it function. So here we have plants, algae, and cyanobacteria, so all photosynthetic. So they have these membranes, and then within the membranes, there's these different complexes that can absorb the energy from photons, and that bumps specific electrons into higher energy states. Now there's a flow, and you can see with this dark line that kind of makes its way through what we call photosystem 2, and then these different kind of carrier molecules that can transfer electrons through the pipeline like a bucket brigade, and then subsequent complexes like photosystem 1. And photosystem 1 was the first to be discovered, which is why we call it 1, even though it comes kind of chronologically later down the line than photosystem 2. Now you'll notice at 2 points here, right, the yellow orbs here and the lightning bolts are showing ways that photons can be introduced into the system to increase the energy states of electrons within photosystem 2 and photosystem 1. In photosystem 2, as the electrons are actually leaving photosystem 2, they need to be replaced. And where they're replaced from as they leave the system is water. So electrons are actually stripped from water molecules and integrated back into photosystem 2 to replace the ones that are lost. And as a result, if you have two molecules of water, right, you can take those two oxygens, one from each molecule, and create diatomic oxygen, right, O2. And now you also, of course, have these four hydrogens, but now they've been stripped of their negative electrons, so they've become positive. So now you have these positive hydrogens, positive hydrogen ions, or what we also call protons, right? Because that's basically what you have if you have a hydrogen atom and take away its electron. All you got left is a proton. So we can call these either hydrogen ions or protons. Now, as they're being concentrated as a result in the lumen of this structure, they're going to be increasingly concentrated, so they're dying to get out. And fortunately, all the way to the right here, right, we have an enzyme that's more than happy to let them out. So this is the ATP synthase, and as it lets them out, it uses that force to phosphorylate ADP to make ATP. And there's also different avenues along this sequence where other hydrogen ions can be also concentrated within the lumen of this organelle. So that's a little bit of the overview of electrons and what they're up to. But you have to be careful as you're thinking through this because there's two actual things that are going on. In one case, depending on what kind of players in the system we're talking about, there's an actual movement of electrons, right? Like a bucket brigade. But within the photosystems, the electrons aren't moving, but the electrons in molecules are bumping up to higher energy states, and that energy is being transferred. So we can look at what we call the antenna complex, or these concentrations of photoreceptive, photoreactive, these molecules that have the ability to absorb photons, right, and bump an electron to a higher energy state. We have these concentrated in these antenna complexes. So what we're seeing here is a blue photon that's being absorbed by a molecule within this antenna complex. It's bumping an electron into a higher energy state, moving it from a ground state into an excited state. And now you're seeing subsequent, with the red kind of bouncing arrows, subsequent electrons in other molecules being bumped into a higher energy state. So it's the energy being transferred, right, not the electrons. And they can make their way around, kind of moving between these different molecules towards what we call the reaction center right here. But if they have the capacity to energize the special pair of chlorophyll molecules, so they're chlorophyll A molecules within this reaction center, then that electron's going to bounce through these different avenues that we saw within the membrane. And you'll get some loss of fluorescence throughout this process, but a lot of the energy will be efficiently transferred from molecule to molecule to molecule. Ultimately, an electron bouncing into a higher energy state will actually physically make its way through some of the subsequent players. So there are the electrons moving. In this second kind of representation and illustration, you're seeing that there's a block. There's some sort of a defect or an inefficiency, or something's going on with this cascade of events within photosynthesis that we cannot excite the electrons within the reaction center. So there's a loss of maximal fluorescence. If you were to look at some of the pigments that are receptive to photons that we see within these reaction centers, right, you can see sun, and there's these peaks of absorption that we're kind of familiar with that can make their way through these different pigments. So there's the carotenoids, there's chlorophyll B, chlorophyll A, and then molecules of chlorophyll A within the reaction center. just generally kind of speaking, they're going to funnel in this kind of direction as electrons are being excited to a lesser and lesser extent because there's energy lost in these transfers. So we see initially some excitation of electrons and transfer of that energy kind of being funneled ultimately towards the chlorophyll A's within the reaction center. So we've seen the illustrations on the top here. Now we're looking at the bottom. What you can see on the left-hand side is one of these Jablonski kind of representations of what's going on with the electrons. And a reminder that initially electrons are in the ground state. So that's an S with subscript 0, right, or singlet kind of orientation or the way that the electrons are represented in terms of their rotational direction. If you remember back to the physics, right, you have pairs of electrons that are moving in different directions. Now, one of these pairs, right, can get bumped up. So that's shown here with the singlet 2 or S subscript 2 is an electron being bumped up to an excited state. And that electron will remain in that excited state, but some of the energy will be lost as vibrational heat energy, and it'll drop down to the S1 state. And you can also see the red arrow pointing up, right? So in some cases, the electron, if it absorbs light of a lower frequency, right? So a shorter wavelength, so a different color, right? It'll be bumped up too. It'll get excited, just not as high, right? And it'll shimmy around in that S1 state. And if you remember, there's kind of substrata within that level. Some of that will also be lost before ultimately kind of settling in the base of that S1 state. Now, with normal fluorescence, right, that electron's going to drop down to the ground state. And when it does that, there's energy being lost between S2 and S1, and even within S2 and S1. So what's emitted is fluorescence of a different wavelength. So on the right, we can see a chlorophyll molecule that's nicely shown these two different absorption peaks in gold, and how nicely that actually correlates to where the electrons within those molecules, how they bump up to the higher energy states, right? Not surprisingly, the shorter wavelength, higher energy photons, right, bump electrons into a higher S2 state, whereas the longer wavelength, lower energy red photons there, right, chlorophyll can absorb those too. It just gets the electrons to the S1 state. Now, one thing to keep in mind is that the fluorescence overlaps here with the absorption. So that might kind of strike a chord with you when we were initially kind of talking about FRET, or this Forster resonance energy transfer, where we're seeing kind of the emission of a photon here, that, or the fluorescence, right? So the emission that can also couple to the absorption or the excitation. So there's some overlap. So we can look at some of these different photosynthetic pigments, and they broadly cover more of an absorptive spectrum or range than any of them do in singularity. So it kind of opens up the range of photons that are going to be able to be exploited, to be funneled into the antenna complex. And there's other pigments that exist too that can broaden this even more. But we see these kind of dual peaks or absorptions within these molecules. Shown on the left, on the top, is chlorophyll A. And then just below that, B, is chlorophyll B. And these are pretty darn similar, right? Except there's an extra oxygen tacked on to chlorophyll B. And based on that subtle kind of chemical nuance that affects the absorption and emission properties of the molecule. We talk about electrons that are being excited. So where are those electrons, specifically what electrons are being excited? We can actually see within what's called the porphyrin ring, there's a magnesium that's kind of bound and settled within that structure. It's electrons within the magnesium that are positioned just right to allow for the excitation of electrons within that atom. And then down below here, we have beta carotene. So this is just another kind of visual representation of chlorophyll A and chlorophyll B. And you can see, right, it might take you a minute to find where the difference is, but there's the oxygen kind of tacked on there that gives the distinction between chlorophyll A and chlorophyll B. So how different is their spectra? Well, we can look at absorption and emission here, right? So absorption is to the left. Again, that should look pretty familiar to you by now that there's these kind of two prominent peaks. But now note, in between 600 and 700 is where you're seeing both kind of excitation or absorption and emission, which, to put it another way, can mean that the photons that are being emitted or released by these pigments will have some overlap, right, with these pigments here, where they can be excited. So this opens up the possibility with these overlaps that there can be this fret or transfer of energy between these different molecules. So looking specifically, for example, we have carotene here. So carotene here can be absorbed by visible light. And the authors in this manuscript cited below use some quantitative analysis to kind of get a sense and get their heads around the percentage of different energies that are moving through these different avenues. So if we have carotene that's getting excited, right, some of that transfer of energies within electrons will be lost as heat. But then with fret, right, almost 3/4 of this fret energy with electrons that are being excited are just moving that energy or transferring that energy then to chlorophyll molecules, right, within the reaction complex, within the antenna complex, within the reaction center. There's going to be the movement of these different energies. So this is pretty interesting and fascinating that you basically have these biological structures optimizing for absorbing photons that can then be in really close proximity to each other, right? So we are looking at kind of what's needed in terms of diameter to allow for the transfer of energy from one molecule to another. So then the authors also present an equation which looks at the extent to which there's efficiency within this fret energy transfer. So R subscript 0 here is defined as the Forster distance. So this is characteristic of two particular molecules depending on what kind of spectral overlap they have. How good are they at partnering up to be able to be involved in this fret kind of phenomenon. And then R is when we're talking about the distance between the donor and the acceptor. So clearly, the bigger this gets, and this is also to a power of 6, there's going to be a significant decrease in the extent to which there's efficiency in the transfer of energy between electrons. So they got to be close. And in fact, some tricky biologists have even used this strategy to be able to measure how close two different molecules are within a cell, because they can come in with a fluorescent protein and kind of genetically fuse it to one protein of interest. So you might get something like protein of interest A that's also fused to a green fluorescent protein, right? But then you can take protein of interest B, and then fuse it to another fluorescent protein. So the cell will express these modified protein A and protein B, two proteins that you're interested in, but these proteins will be fused to fluorescent molecules that have the appropriate characteristics, like the Forster distance and so forth, that one theoretically could excite the other, right? If the emission and the excitation are compatible, but only if the two proteins exist within close proximity to each other within the cell, right? Or else you get a failure to allow FRET to happen. So it's actually one of my favorite fluorescence techniques, where you can use this capacity to kind of understand the physical distance between two biological molecules in a live context. So we've been looking at different ways that the energy of photons can be used to excite electrons, and how electrons that are excited within these different pigments can kind of transfer this energy from one pigment to the other. So how can we actually measure, so this is different but still dealing with some kind of quantitative aspect here. How can we actually measure how well photosynthesis is actually progressing? So if you're interested in measuring photosynthesis and you want to quantify photosynthetic efficiency depending on some experimental variable or some context, right? You can measure the uptake of CO2, right? So this will be a reflection of fixation, so how much of that CO2 is being integrated into the building blocks of sugars. That's what in large part photosynthesis is all about. So we can measure the uptake of CO2 or measure the production of oxygen. So remember, early on in photosynthesis with the light reactions in photosystem 2, you have electrons that are being evacuated or leaving photosystem 2. So we have to replace these. So where do we get the electrons from? We get them from water, right? So we split water. And as a result, we're taking away the electrons. So off they go are 4 protons, 4 hydrogen ions, missing their electrons. And you're kind of left behind with 2 atoms of oxygen that double up, so that they each have the 8 outer electrons in their outer shell. So this is diatomic oxygen. So we can see how much of this is being produced. Okay. We can also just see how much bigger the plants are. So this is using the expectation that the energy from photosynthesis, apart from respiration, is being used to build sugars or molecules that are using those sugars. So just an increase in just weight, right, or material should be a reflection of the amount of photosynthesis. And then also DCPIP reduction is a technique that can be used to see how many electrons are being freed up through photosynthesis, because that'll affect colorimetrically this reagent, so you can see visually or with a spectrophotometer how much photosynthesis is going on. So with the first one, there's an instrument that's an infrared gas analyzer that can measure vibrations of molecules within a gas. So it's taking advantage of some signatures of CO2, where you can actually in real time measure the amount of carbon dioxide that's being taken up by living plant tissue. So these data here are showing on the bottom the intensity of photosynthetically active radiation, right? So that's light, excuse me. And with increasing light, how is that affecting photosynthesis? So the authors here look at this species of plant at a particular temperature, and then they look to see if you preemptively kind of give them high or low temperature or high or low moisture, how does that affect photosynthesis during the quantification trial or period? And they can plot this. So looking at different types of temperature or moisture kind of paradigms, how is photosynthesis progressing during the measurement period. That's measured here on the y-axis using one of these instruments. So you can measure how much CO2 is being used, right, or how much O2 is being produced. So this is pretty straightforward, where the gas being produced is through photosynthesis and is oxygen. So you can use the amount of gas being produced to mechanically drive something like an instrument like a syringe, which can collect the gas, right, and it moves to the degree that gas is being produced. So this is another measure of photosynthesis, or a way that you can quantify it. So you can also look at increase in dry mass. So these authors here looked at basically different colors of light, right? So here we see the different spectra up here. And then they weighed, right, looking at these different spectra and these different intensities. What was the effect on basically dry mass is what is kind of interest here, right? Because again, there should be a reflection of photosynthesis, because photosynthesis fixes carbon to integrate into organic molecules, which would from which you would derive the dry mass. All right, so DC PIP looks like 2,6-dichlorophenolindophenols is something that can be modified in the presence of protons and electrons, right, which should be produced by photosynthesis, which change something of a blue color into a clear color. So normally chloroplasts are freed up by homogenation and filtration and then put into a solution that includes this reagent, and then the reagent will change color, so it's colorimetric, based on how much photosynthesis there is, right? And we're using the production of hydrogen ions and electrons, right, as a proxy for the amount of photosynthesis that is occurring. Okay, so this was another kind of delve into photosynthesis, looking at FRET, the way different molecules within chloroplasts, more specifically within photosystems, more specifically within photosystem 2, right, how they're absorbing the energy from photons and transferring the energy between molecules. And then we looked at specific ways that we can quantify or determine how much photosynthesis is occurring within photosynthetic tissue. Here are the references from the couple of the studies, a review, and then also an analysis of FRET within plant tissues from the presentation.